Hello student, very good morning to all of you. How are you? I hope you all are happy, healthy and fine at home. Do you remember what were we discussing yesterday? I think we were discussing about the synthesis of glycosides, and in that we have also discussing the donor acceptor concept regarding the formation of glycosidic linkage or glycosylyl bond formation. So that type of the thing we have seen. Glycoside bond formation generally require activation of the sugar at the anomeric center, and as we know that anomeric center is nothing but where the glucose can shows as a different absolute configuration. One is rotating the plane of polarization through right hand side, another through left hand side, and therefore that type of the conformation of are known as anomers of each other. That type of the diastereomers are known as anomers of each other. And the glycosidic bond formation, how that bond formation takes place? While formation of the bond, glycosidic bond formation, there must be one should be as a donor and one should be at acceptor. In nature, then and they come together due to the special force which is known as the electrostatic force of attraction, and the bond is formed, or sometimes it is referred as a O glycosides. O glycosides means there is a formation of the bond and elimination of the water molecule takes place over there, and there is a linkages occur. CO, or that is ether oxide linkage is formed, so that is also referred as O glycosides, or the synthesis of oligosaccharide. And as we know that oligosaccharides are further at the beginning, while studying as a classification of carbohydrate, there we have seen carbohydrates are mainly divided into as a three subtype. Number one is a monosaccharide, second one is a oligosaccharide, and third one is a polysaccharides. And monosaccharide do not hydrolyze further by using the what we can call it as acids or specific enzyme, but the oligosaccharide further divided into as a three type. Number one is known as a disaccharide, second one is a trisaccharide, and third one is a tetrasaccharide. These are the sub classification of the oligosaccharide, and where you can see or where you can observe or where you can notice the O glycosides, that bond formation occur, and there is a removal of water molecule. So the glycosidic bond formation generally selectively occur at the anomeric position, anomeric hydroxyl group. Generally sometime in some books refer as a anomeric hydroxyl group at 2 position, or sometime it is referred as a 1, because if you consider as a heteroatom as a 1, then it comes carbon number first, it comes at 2 position. If you see only as a starting from the anomeric carbon, if you referred as a 1, so that is a carbonyl carbon. Also there are 2 possibilities are there for. So this type of the discussion we have discussed in our previous lecture, and as I already told you that this is a very significant or the prominent as far as the research point of view is concerned, and therefore University Savitribai Phule Pune University introducing this topic at a M.Sc second organic chemistry, and that's why we are studying this. Is a special and the very interesting subject as far as carbohydrate chemistry is concerned, because carbohydrates are easily, or they are the natural product, easily available or cheaply available we can, and starting from that there, and most of them are optically active, and optically active we know that those compound has a chiral center. They are optically active in nature, even though there is a some points are there, some type of the symmetries are present in those organic compound, like as a plane of symmetry, alternative axis of symmetry, or center of symmetry. Then that compound turn into as a optically inactive in nature, but those are not possess any kind of the symmetry. Just now I mentioned one is a plane of symmetry, center of symmetry, or alternative axis of symmetry. Those symmetries are absent, then and then that compound definitely is optically active in nature, so it can rotate the plane of polarization either right hand side or left hand side, depending. This is the experimental quantity, or what we can, this is a physical property of that organic compound, where we can measure these type of the properties. So in the last lecture also we have discussed synthesis of oligosaccharide, like as beta glucosides, beta galactosides, and alpha raminosides, etc. 1,2-trans glycosides, beta glucosides, and beta galactosides, beta xylosides, and alpha mannosides, alpha raminosides, etc. are usually prepared with glycosylyl donors containing participating neighboring group, or we can call it as a neighboring participating group, acetyl benzoyl, pivoylyl, etc. at 2-hydroxy, and some example for gal B, gal B, gal L, and L alpha gal C are we already discussed in scheme number 2. And instead of the methyl glycosides of azido galactose, the corresponding glycosylyl fluoride has also been used as a acceptor, and in general glycosylation yield are high irrespective of the anomeric configuration of the trichloroacetamate. We know that trichloroacetamate, it is act as a donor here, and only one anomer is obtained occasionally when glycosylyl acceptor with more than one free OH group are used. High regioselective is obtained because of the different reactivity of the OH group, and it was found that dichloroacetamate donors are specially appropriate for deactivated fluorine containing donor in contrast to the other glycosylyl donor thioglycoside we will see in a future. A highly convergent synthetic method has been developed for the synthesis of cello-octose and other cello-oligosaccharide via di- and tetrasaccharides, which can be converted either into glycosyl donor by dealkylation and transformation into the trichloroacetamate, or into acceptor by deacetylation. That scheme number 3 also we have discussed in our previous. So today right now we are going to discuss the use of paramethoxy benzyl group PMB instead of acetyl group was less successful. The combination of the acetyl, pi-vinyl and benzyl group leads to the proper balance between stability and the reactivity of trichloroacetamidets as a donor and the acceptor substrate. High yield were achieved by using a specially developed high vacuum system for glycosylation under anhydrous condition. Trichloroacetamates as have also been used for the glycosylation of more complex carbohydrate acceptor, that is the scheme number 4, which we are going to see shortly. Gal-B, Gal-C and other disaccharide were synthesized and converted into a fluorescein labeled acceptor, which are used for spiralyl transferase arrays. Glycosyl dioxazine has been used as a carbohydrate acceptor for further glycosylation with various trichloroacetamate, that is the scheme number 4, and that we will see. Right now all glycosides were obtained in high yield and high stereoselectivity because of participating neighboring group at 2OH in the synthesis of dissociin, polyphenylene, D-balatin 7 and gasoline, and that the scheme number 4th we are going to discuss over here, and scheme number that is also belonging to as a trichloroacetate. As we know that trichloroacetamates, the structure of trichloroacetamate is like this. Let's see over here. This the structure of trichloroacetamate. I am showing that structure over here. Here is O, then here you can write double bond NH, then CCl3. At 2 position there will be acetyl OAC, that is a protected OAC, also protected over here, and one at axial position OAC, and here OAC we can protect over there. And now this is a general compound, and when this compound will be treated with this compound, let's see over here. And now remember this, I am darkening this bond, it means that they are above the plane. This is the general thing you have to remember throughout these type of the glycosidic synthesis of glycosidic. That time you remember that one, they are projecting above. Now the another compound which we are writing over here is this one. Now the same compound I am showing over here. This is at equatorial position. One is O, then another bond is CH2 twice 8 in that C, then CH2 twice 8, then there will be NH CBZ, then NHCBZ, that is a carboxylic group, that is a protected, that amino protected group is over there, and then NHAC. Here you can write NHAC acetyl group. At equatorial position there will be hydroxyl group, which is a free hydroxyl group, and here at there is a oxygen atom, then you can write this one, there will be oxygen atom, and then there will be pH, that is a protected with that not benzaldehyde here just showing as a one methyl group here, another methyl group. When this compound and this compound, you observe carefully, they get reacted together in presence of first one with, that is BF3, as we know that BF3 OET twice, that is a trifluoro boron etherate in presence of dichloromethane CH2Cl2, and the temperature is kept here is not temperature at all, but the yield is obtained, 76% yield is obtained. And when it got as a 76%, this is further treated with one deprotection. Number one is a deprotection. Deprotection means deprotected that protecting group, and second one is a fluorosine isothiocyanate. Fluorosine, second one is a fluorosine isothiocyanate. This is the reagent adding for deprotection, and what you are going to get over here, the compound which you are going to get here, remember here the compound which we got over here, this one I am showing over here, then oxygen, then another one where we are showing this one oxygen atom over here, this compound, and further at equatorial position C, CH2 8 times, then NH, it is a NH, then there will be double bond yes, there will be double bond yes, and then here is a NH, then this compound further shown by this compound here, CO2 Na at meta position, CO2 Na, that is a carboxylic group, sort of that carboxylic group, and at this position there will be three membered ring again attached over here like this. This is the ring, not here. Here is the oxygen atom, here is not oxygen at all, here double bond O, then at this position double bond, here also has a double bond, they are in the conjugation. On this side there will be a this type of the conjugation and hydroxy group over there, and this the conjugation, highly conjugated compound is obtained, and here what you have to show over here, all that get deprotated. Now I am darkening, or this thick bond clearly indicate that is above the plane, and this darkening, it means that they are above the plane, and other part is that goes below the plane. Here all are the deprotected group, here hydroxy group, all are at equatorial position. I am showing not, this is at axial position, not equatorial, here is at axial position, and this hydroxy group again at equatorial position. So this is the compound which we got here, again hydroxy group at equatorial position, and now here the hydroxy group will be shown represented like this, and here is a NHEC, here again as a NHEC. This is a protected group. So this is the compound with high yield, near about is more than 70% of the yield is obtained over here by combining this compound. So this is the what we can call it as a shown one of the scheme, that is the scheme number 4 is represented over here. Now another compound we can show, there are the various different combination we can combine together and we will get the different type of the molecule. Let us see we can combine which of the compound we can combine over here so that we can get this. Let us see here, this is one of the scheme, this compound and this compound reacted together in presence of trifluoro borane etherate in presence of CH2Cl2 dichloromethane used as a solvent, and this whatever intermediate they have got over here, this compound again carried out as a deprotection process in presence of fluorosin isothiocyanate, and they fluorosin isothiocyanate, and they get the compound in a high yield. So that is the things which they have got over here. Now let us see, in addition to this, another the compound which how we are getting that high yield product. Let us see that also here. Let's see, suppose we have this compound, let us see this one, oxygen atom over here. So I think we made something is not like this, it must be as a chair form. In a hurry I am shown like this, but remember this is the compound, here the oxygen atom, and then we can draw here double bond NH and CCl3, that is a trifluoroacetimidates we can draw over here. And always remember this is a thick line represent the molecule above the plane and other goes below the plane. Here OAC we can write over here at axial position OAC, and at this position we can again write OAC. At equatorial position we can write OAC, and here at equatorial position we can write methyl group here. So methyl group CH3 methyl group. Then another molecule we can draw over here. This is a fructose type of the compound we can draw over here, and here we don't know this zigzag this type of the arrow clearly indicate that it is not the definite whether that bond is above or below the plane, that is not confirm, and that's why it is shown as a like the CCl3, that is a trichloroacetimidate we are trying to write over here. And this also we can show as a thick bond clearly indicate this is a above the plane, and where here is a OAC we can write, and here again at this position, at two position we can replace that is above OAC, and here you can write the bond like this, the OAC, all are the protected only, this and this. Among these two, one is act as a donor and one is act as a acceptor over here. Then oligosaccharide we can represent here also like this, O, then this one, then oxygen, and then we can represent this compound like this. This is a compound we cannot here is a this way oxygen atom we can show here, and then this will be shown like this, and then further this is not again azine, double bond NHCCl3 trifluoro acetimidates. Then here you can write OAC. Here at this position we can write again as a OAC, and here at equatorial position we can show the protected acetyl group, and see this ring, observe this ring carefully, again OAC. But remember, this thick bond clearly indicate that they are above the plane, right? This one, and therefore here you can write OAC, and here you can write OAC, here also OAC, and then OAC. This type of the compound you can represent, and after that you can treat with this the compound. I am going to show over here this one, another one. This is the compound where you can show at axial position there will be NHCCl3. All are the trifluoro acetimidates, and again this is a thick line, then here OAC, and again over here OAC, and here we can write OAC. All these things, when you bring all this together, you may get the compound like this compound, this type of the compound. You may end up with this is the compound. Here again thick line, here must be OH, then we can show O here, and then this is a thick line, and then 6-membered ring you can show over here. Another 6-membered ring you can show over here. Another ring where is a double bond, the double bond in the another ring over here, and one the bond which is above one, then 6-membered ring again join over here like this, and then there will be a 5-membered ring, and 5-membered ring should be joined with 5-membered ring should be joined with again here is a 5-membered ring, and after that there will be a here oxygen atom, then this one, this is not this one, the 6-membered ring joined over there, again it is continued till there. So here we can write O again, this is above the plane, so here OH we can write, and then there will be oxygen atom, then here oxygen atom, and this here is a pH, that is a benzaldehyde protected type of the group over there. Here also that is a stereochemistry shown by this methyl group, and one methyl group may be here at this junction that is above the plane. So this is represented as a scheme number 4. So in this way we have seen some of the example with the help of donor acceptor concept glycosylyl formation, that is a glycosylyl bond formation, that is a donor acceptor concept, by using as a trichloroacetamate. In next time we will see some of the example regarding as a thioglycosides and some of the halides, what they are, whether they are acting as a acceptor or donor, that type of the concept we will discuss in the next lecture. I think with this, let us stop today here. The remaining we will see in the next lecture. Thank you. Have a nice day.